I think we'll begin, and hopefully the sun will come out in a minute and hundreds of people will suddenly reappear. Okay, so today's session is different from all of the other ones that we've had over the past 11 years, each one of which has been designated or designed to talk about a specific disease or disease process from the standpoint of its translational opportunities and the basic science that underlies it. And as you know, most of the time there's a live patient who puts a human face on the disease. But today's discussion is going to be a one person, and we're very, very fortunate to have Christine Grady be that person, because the topic is one that permeates everything we do. So it's great to discuss whatever it may be, cancer or some infection or something like that, from the standpoint of how science is needed and what's needed from the clinical standpoint. But often we lose complete track of the fact that we don't operate in a vacuum just with our own test tubes. I guess nobody uses them anymore, so our own gels. And so the issues of ethical considerations permeate the whole spectrum, from a research enterprise at a bench to a clinical enterprise at a bedside and beyond. And this is very serious and provocative material. So we're really very fortunate to have Christine Grady, who is the clinical center. She's the director of the clinical center department of bioethics. Christine went to nursing school at Georgetown, and then studied in Boston, and then obtained her PhD in Georgetown, and became increasingly interested, involved, and is a national and undoubtedly international leader in thoughtful consideration and analysis of the science of ethics. Ethics is a science, and it bears examination and critical assessment, and remarkable things come out of that. So we're very grateful to you, Christine, for coming over and speaking to us. And the format, particularly for those who haven't been here before, is very informal, and particularly with a smaller group than usual here today, although I suspect there are several hundred who are watching online but don't want to get their feet wet. So we encourage you to raise your hand, stand up, ask questions. You can save it to the end, or I'm sure that Christine would welcome questions during the course of her presentation. Okay, so welcome.
So thank you very much for that introduction. I not only welcome questions, I'm going to ask you some questions. So hopefully you'll stay awake with me. My task today is to talk about ethics and translational research. Two huge topics, very difficult to really compress into an hour or so. And so I suspect that at the end of this time together, you will maybe be demystified, but you'll probably, I hope, actually leave with lots of questions swirling around in your head that you'll be thinking about for many days after today. So to start with, I'm speaking for myself, not my department or the NIH, and I don't have any conflicts of interest. And what I hope to do in the time that we have is speak a little bit about the spectrum of biomedical translational research, the goals and norms that attend to that, some of the ethical constraints and frameworks that are useful in thinking about it, and then talk about some cases and end with a few examples, of how ethics has been useful in facilitating translational research. So that's the goals, and we'll see how we do. So to start with, everybody, I love this quote from this paper from Stuart Wolf in 2008, where he says, translational research means different things to different people, but it seems important to almost everybody. And I think it's really interesting if I asked you, what do you mean by translational research? Or if you walked down the hall in your respective workplaces and asked, you'd probably get different answers. But at least it encompasses starting with a research idea that's designed to help benefit patients and the public health. So it's that translation from the ideas to something that's useful and is going to improve the public health. Now, some people have divided it into two, the T1, T2 universes, if that's right. One is the bench to bedside part, and the other is the translating the research findings into practice. I've also seen models, I think I might actually have one, that have put T3 and T4 on that list, so you can divide it up in different ways. But there was a clinical research roundtable effort in the early 2000s that first began to talk about what they called the translational blocks. And they pointed to, this was from the Institute of Medicine, I may have said that, they pointed to the T1 block, which is right here, between basic science research and clinical research, and noted that that's really primarily the domain of NIH. And then pointed to a second block, which occurs from the findings of research to other kinds of implementation research that make it, effective in practice and in public health. And NIH does fund some of this as well, but the bulk of NIH really is there. This is just another model that has more T's than the IOM, but I'm not going to really go through it. But it gives you the idea that there are different stages in this translation, but they're all important to think about in terms of how we get from an idea to something that's going to help a patient. Now, the NIH, one might argue, is basically devoted to translational research in general. The motto is turning discovery into health. So everything we do here, from the basic science all the way through, is to translate research findings into something that's going to help people with illnesses and the public health. And I've seen it written, actually, that the model that the clinical center has set up, that the NIH intramural program has had a commitment to the bench-to-bedside part of the translation pathway from the days that the clinical center opened in the 1950s, because it was deliberately built in a way that patient care units and patient beds would be in close proximity to the sort of leading-edge laboratories, and so there would be this very easy bench-to-bedside translation. So with that sort of very brief framing, I'd like to talk about ethics now for the rest of the time. And certainly it's worth remembering that the goal, again, is to develop socially valuable or useful knowledge that can be translated into better preventive, diagnostic, and therapeutic interventions to improve health for people. And I think it's, again, very important to remember that it comes on a spectrum, and that every part of the spectrum is part of the translational process, and an important part of the translational process, and a part of the translational process which has some ethical considerations that need to be attended to. So there was an interesting paper, and I provided it to Dr. Arias. I think it's available now on the website. Is that correct? That Steve Jaffe and Frank Miller wrote a couple of years ago, called Bench to Bedside, Mapping the Moral Terrain of Clinical Research. And I think it's a very useful paper to thinking about, to help think about the ethics of translational research from across the spectrum. And they made the claim that there are three dimensions that form the basis for ethical responsibility and deliberation in research because of the morally significant consequences for society, science, participants, and the whole research enterprise in itself. And those three dimensions that they described were the ethically appropriate goals, the internal norms, and the ethical constraints. And they pointed out that the first two, the goals and the norms, are pretty uniform across the spectrum. In other words, the goals are pretty much the same, whether you're in the laboratory or in the field or doing implementation research. The internal norms are also pretty much the same across the spectrum. But the ethical constraints vary depending on what stage you are in the spectrum. And so I'm going to talk about each of those briefly in turn and then turn to some cases. So the goal, of course, is whether conducted in the laboratory or the clinic, the pursuit of generalizable knowledge that has the potential to contribute sooner or later to improving health. Yeah, that we already talked about that. And then they talk about that part of this is driven by a moral concern for promoting health and reducing suffering. So going back to sort of basics, why do we do research? We do research to find ways to understand health, to improve health, to reduce suffering. And so with that always as the central goal, it helps to shape the kinds of things that we determine are okay to do and not okay to do based on, in pursuit of this goal. They also point out that recognizing that we want to reduce suffering and promote health helps us to make difficult sometimes decisions about allocation of resources or using resources for research in a way that commits them to the socially valuable research, and that's an important consideration when we're deciding how do we allocate research monies. And then the other thing they point out is that the potential of a proposed experiment, a particular experiment, to realize the goal of improving knowledge is necessary in order to justify any kind of risk to either animals or to people in the process of research. They also then point to the second dimension they point to are internal norms. And they say that there are internal norms in the scientific enterprise, again, across the spectrum, that are intrinsic to the process and necessary to ensure the trustworthiness of the enterprise in itself. And there are two sort of sets of internal norms that they point to. One are the norms of the methods, of scientific investigation, and the other are the norms of scientific integrity. These are not new to anybody in this room, but they're important to sort of put on the table in terms of thinking about how this all fits together. So this is a complicated table, which I'm not going to go through. It's from this paper. But basically what it does is take the development of linezolid for treating MRSA and start with the research question hypothesis, the experimental materials and conditions, the intervention that was used, and the outcome measures, or the devices that were used to measure, and follow it along the spectrum of laboratory science, animal science, research with healthy volunteers, and research with sick patients. And what their point was here was to try to show that the methods of scientific investigation are relevant to every stage of the spectrum of translation in research. Then they briefly point to the norms of scientific integrity that are, again, central to research across the spectrum. And I know this is not a talk on scientific integrity, but certainly we all recognize that there are very specific definitions of scientific misconduct. And I think we also can recognize that scientific integrity is much more than avoidance of fabrication, falsification, and plagiarism. And that's been recognized by a number of people over the course of time. And what are those norms? Familiar norms like honesty, accuracy, efficiency, objectivity, competence, collaboration, transparency. I'm sure there are others. But again, these are norms at every stage of the scientific spectrum, the translational spectrum. These norms adhere. And yet, we are aware that there's some, that they're not always adhered to. And so I love this cartoon. I want to change the career in politics to a career in ethics and ask the question, you know, a career in research, sorry. Is ethics integral to a career in research? Does every person that does research have this sort of as part of their fabric? Or is it something that they drop at the door? Or is it something, and I think increasingly it feels this way to me, where researchers feel like it's a burden that they'd like to hire somebody to take care of. They want to outsource it. You know, whatever the ethics is, it feels like burden and regulation, and it's not really integral to what they're trying to do in their science. And I think that's really a shame. What gets in the way of the norms of scientific integrity? Well, certainly, you know, easy to recognize things like people who haven't been sort of taught what the norms are, sloppiness sometimes, hubris, I think, is a huge one, and increasingly, and I think people are worried about this, competition. You know, competition for resources, competition for money, et cetera, makes people want to cut corners and not integrate ethics into their daily activities. What's interesting is competition, although the pay lines are going down, the competition is certainly not new in research. And I found this quote by Walker Percy from a book that he wrote in 1971, which I love. It says, "Lord, grant that my discovery may increase knowledge and help other men. Failing that, grant that it will not lead to man's destruction. But failing that, grant that my article in Brain is published before the destruction takes place." This is kind of funny, but not so funny. So the third dimension that Jaffe and Miller talk about is ethical constraints. And so this is the place I'd like to spend the bulk of the time that we're talking together. And again, it goes back to recognizing that research at every stage of the spectrum has consequences for society, for science, for the people that are involved, all the people that are involved in it, and the enterprise in itself. They recognize, for example, that without this dimension of research, in other words, even with the right goal and with some of the norms, the internal norms, somebody could conduct research, but it wouldn't necessarily be ethical research, and it could be unethical research. So understanding and attending to the constraints are an important part of how people should conduct research. Again, they give us a chart which is more complicated than I want to focus on, but I just want to point out a couple of things. That the ethical constraints, if you look at them, all incorporate issues around risk and benefit, common constructs. So the laboratory science ethical constraints are minimizing external risks and pursuing beneficent purposes. You add that to that when you're using animals, reducing burdens and harm to the animals. You add to that when you're using healthy people, avoiding unacceptable risk, exposing people to unacceptable risk. And you add to that when you're using people who are sick in research, avoiding risks of omission. So what that means is not doing something for them that you should be doing based on their illness because of the research. So you're not only not exposing them to risks by procedures, et cetera, but you don't want to not treat them in the way that they ought to be treated. So let's take an example. This is one that probably is familiar. It's been in the news recently, and it's got some really interesting, I think, issues that go back to these goals, internal norms, and ethical constraints in a very nice way. So this is the H5N1 avian influenza experiments. As you probably know, H5N1 is a major threat to the world, public health and agriculture. Something like, I forget the number, millions of birds have been culled because of avian flu. There have been about 600, only 600 documented human cases, but of those 600, it's about 60% fatal. So it's a pretty serious infection. And you may recall that about I think it was early 2011, there were 2 gain-of-function studies that were done that showed that in the laboratory they were able to mutate the virus in a way that made it more transmissible. And they were able to show that it could be transmitted from a ferret to another ferret through respiratory droplets. So these two experiments led to lots of concern about the possibility of accidental release or nefarious use of these mutated viruses. And there was an editorial in the New York Times of January of 2012, which was pretty dramatic, I thought. An engineered doomsday. They pointed out that these experiments funded by the National Institutes of Health have shown in a laboratory how that could happen. In the process, they created a virus that could kill tens of hundreds or hundreds of millions of people if it escaped confinement or was stolen by terrorists. So this led to, in the scientific field of influenza researchers, a voluntary moratorium and lots of intense discussion, meetings all over the world about this issue. And the global discussion really was centered on the benefits and risks of funding, conducting, and publishing these studies. And so it really goes back to this ethical constraint in laboratory science of figuring out ways to minimize external risks and while pursuing, if that's the right way to think about it, beneficent purposes. A lot of the discussion was also about scientific independence and transparency. What are the norms of science? Science is normally transparent. Scientists are normally allowed to conduct their research without people telling them you can't publish it or you can't do this or that. And so the debate centered on this. And as some of you probably have seen, just last week, I think it was, a framework was published by people here at the NIH on how decisions will be made about funding research of this type. And if you look at the criteria that they spelled out, and I'm not going to read through each of them, basically the criteria are trying to do the two things that the ethical constraints were. They're trying to find ways to minimize external risks and to make sure that the experiments are being done in the pursuit of beneficent purposes. And there are lots of details in here about how they might go about doing that, but that's really what they're trying to do. So now going back to this very large chart, I want to turn to some of the issues in the bottom half of the chart about clinical research with healthy people or sick people, and think through some of these constraints in the context of some cases. And again, remind us that the sort of goal here is to always try to promote benefit through research while protecting the participants that are part of the research. And again, that's the sort of the goal of translational research, countered by or sort of qualified by the ethical constraints. So some years ago, some colleagues of mine and I put together a set of criteria that we thought were very useful in thinking through what makes clinical research ethical. And they mirror some of the ideas that were in that other chart. But let me just run through them very quickly because I think they will resonate with many of you in terms of how to think about research. At the beginning has to be a question that's worth asking. That's going to give us an answer that's useful, that's going to be used in the end to promote health in some way. Once you have a valuable question, the study has to be designed in a way that you'll get an answer to that question that you can then understand the answer to, an interpretable answer. There needs to be many considerations in terms of selecting the subjects who will be participants in that research and the sites who will be participating in the studies. And those include scientific reasons and fairness reasons, as well as concerns about risks and benefits. Risks and benefits then need to be, risks need to be then further minimized and then justified by the potential benefits. There's independent review in the process. In our system, it's usually through IRBs, but lots of other review bodies as well, often informed consent. So only after you've gone through all these other things do you approach people and say, would you like to participate? And the informed consent process is complicated in many ways, but it's a very important protection for individuals to be able to say yes or no, I do want to do this, based on understanding what it's about and how it comports with my interests. And then there's a whole host of things that are necessary to do once a study starts to respect the people who are in the study, both during the study and after the study is over. So here's a case that you may, again, recently found out about, I guess is the way I would say it, although it was done a long time ago. It was done in the 40s, but it was only uncovered in around 2010. And this was a case of STD prophylaxis that was done in Guatemala in the 40s. And the goal of this study was to, as written, to understand or to test methods to prevent syphilis. So this was after the war. Penicillin was available. Treating syphilis was now possible, and everybody was very happy to have a treatment for syphilis. And so now the question was, how do we prevent it? So this group, among other experiments that they did, proposed and conducted these experiments in Guatemala, which involved intentional exposure of participants to not only syphilis, but also gonorrhea and chancroid. They were looking at chemical prophylaxis, and they were looking at natural history of these infections. They used methods of intentional exposure, originally planning apparently to use what they called natural exposure, which was taking infected prostitutes to the prison and seeing how many prisoners developed syphilis from having sex with the prostitutes in the prison. When they discovered that the transmission rates in using that method were lower than they had hoped, they sort of used increasingly invasive means of giving people these infections. And what's interesting and disturbing about reading the records is that it appears as if there was a sort of escalating sense of desperation in terms of getting a method that worked in transmitting some of these infections. So there were skin contact experiments and direct injection, scarification, and even cisternal puncture, which is like a lumbar puncture, but in the cistern in the back of the brain. And the people that they used in these experiments were commercial sex workers, initially only as vectors, but later they were also inoculated with some of the STDs, prisoners, soldiers, and residents of a psychiatric hospital. So using this kind of a framework, I think it's easy to see that an experiment like this is problematic ethically for a number of reasons. But starting at the top, I mean, you might say that finding methods of preventing STDs, preventing syphilis, is a reasonable goal. Beyond that, they sort of flunked everything. If you say, were the methods that they proposed or used valid ways of trying to achieve their goal? And the answer is probably not. I mean, there was no good rationale for the methods that they used. There was no good documentation. There was no logical sequence. And in the end, actually, no reports of what they did, no publications. They used only vulnerable subjects with no rationale and no protection. They neither minimized nor justified the risks that they exposed people to. There was some review, but there was evidence of deliberate secrecy about the methods that they were using and what they were doing in general. There was no informed consent, and there was no respect for the individuals who were infected in these experiments, in the sense of when they were infected, how they were treated after they were infected. So contrast that, for example, to a more recent study, which was also an STD prevention study, if you will. This is the HIV Prevention Treatment Network 052 study, which was a real game changer in terms of HIV. which was able to show pretty definitively that treating a person with antiretrovirals protects their sexual partner, at least in a serodiscordant couple. So again, you have the importance of finding a way, an effective means of prophylaxis for HIV, a rigorous design that had careful justification, sample size, etc., carefully selected competent adults in 9 different countries, lots of monitoring and testing, lots of interventions to reduce the risks, at least one IRB review at every site, and lots of other review bodies along the way, attention to informed consent, both written and oral, in the local language, and regular follow-up and testing, early release of the findings because of the DSMB finding that the benefit was so great. So you might say that this is sort of a model of a study that was done in an ethical manner. And yet, interestingly, the PI of this study and some of his colleagues wrote a paper just a couple of months ago where they describe in some detail what they call the ethical odyssey. And I think this is worth spending a minute paying attention to, because if you think about it, they had a good question, a good design, very careful attention to the rights and welfare of the people in the study, and yet there were lots of ethical challenges that they faced along the way. And they just described a few of them. One was, study design. So even though there was no definitive study showing that if you treat one person you can prevent transmission to another, there were many people who challenged that notion because there were observational studies and other kinds of studies that at least made people believe that that was the case. Site selection was challenged because they needed people at high risk in order to show the effect of prevention if it worked. But in those settings, offering ART to people who had more than 350 CD4 cells was not the norm. And so there was a question of whether they were sort of coercing people or incentivizing them to participate in ways that were not reasonable. The prevention package and all prevention studies, and HIV prevention studies anyway, this is a huge discussion because there's a ethical norm about giving people information about the interventions that are known to be successful at preventing HIV, and that's in tension with, if that's the right way to think about it, being able to answer the question, the study question. In a fast-moving field, the guidelines change, and that creates interesting problems. And then there was the question at the end of the study, how do you think about providing or continuing to provide as a prevention intervention a medicine to people who were in the study that's not available to people in that same country who are ill with HIV, or it's not available at that CD4 count. So these are interesting challenges that they faced along the way, which made me want to sort of talk to you about how even within the ethical norms and the ethical constraints that we recognize, there's a lot of judgment to be made and a lot of challenge in terms of figuring out the right way to go. So let me give you a couple of examples. The one thing that comes up all the time in clinical research is the use of placebo. When is it justifiable and acceptable to use placebo? And this is a good example of, in some cases anyway, exemplifies the tension between wanting to find a rigorous answer to a scientific question that will help people and protecting the individuals who are part of any particular study. And so one example that got a lot of attention was a double-blind randomized control trial of St. John's wort against sertraline for major depression. And the trial was designed with a placebo arm, and it got a lot of criticism for that reason. Major depression is a serious illness, and the question was, is this a risk of omission that's tolerable and acceptable? And yet, there was, the tension of how do you really know whether St. John's wort, which is what they were trying to study, works against depression unless you include a placebo arm. And the results of this particular study at the time, anyway, sort of vindicated, if that's the right way to think about it, the people who made that argument, because in the end, neither the St. John's wort arm nor the sertraline arm were any better than placebo in the trial. So what does that mean? It means St. John's wort doesn't work, but it works as well as sertraline. Does that mean sertraline doesn't work? I mean, and subsequent trials have tried to sort this out a little bit more, but in this case, the use of placebo was very helpful. There are some interesting questions about what fair subject selection means in a certain case. So this is a study of a malaria vaccine, the kind of malaria vaccine that would be given to people who are already infected to prevent them from getting serious disease. So not to prevent infection, but to prevent serious disease. And so this kind of vaccine would be very beneficial for people in countries where malaria was endemic, but not beneficial to travelers or military people or people who might be exposed to malaria. So the question was posed in this case, what's the right population to study for a phase 1 experiment of this vaccine? Is it healthy volunteers in the United States, or is it healthy volunteers in Mali in this case, because Mali was a collaborator and has a large issue with malaria? What do you think? What do you think is the better population? Mali. Why? Okay, they might benefit most, but note it's a phase 1 experiment, meaning phase 1 experiments testing tolerability of the vaccine, safety, no expectation of benefit from the experiment itself. I mean, you still could be right. I'm just pointing that. You still think it's Mali? Yeah.
I agree. I think it's Mali because if you are going to eventually
Okay. Sure. Again, important consideration, but in this particular study there would be no controls. It's a phase 1, so it's small number of people, maybe 20. First in human, we just want to know if it's safe.
Right, but there should be differences between the U.S. population and the population.
Okay.
That might influence.
Okay. So you think even safety might be better informed by doing it in people who might ultimately use it? Yes.
Would you sort of repeat the comments so people...
Yes. Oh, sorry. Yes, I can. Yes.
I would actually say both.
Yes.
I would say both.
Okay. Can you say why? So the response was both. Since it's a phase 1 study, the answer is both. Go ahead. Yeah? Okay, so the point was made, maybe there's no reason to pick one versus the other. No ethical reason, no scientific reason. Is that a fair restatement of what you said? You didn't use those 2 words, but I'm putting them in your mouth, yeah.
There are certain places where people have genetic differences. People that are positive for thalassemia have some resistance to malaria, so that would be a question I have.
Okay, so the question is, there are certain places in the world that have resistance to malaria, for example, from thalassemia, and so the question would be, is that relevant in this case? In either the U.S. or Mali, I guess, yeah. Okay, so in this case, I mean, as you can see, I'm putting the references for all of these at the bottom. There were a lot of interesting issues that got raised, and I want to, again, bring us back to the sort of ethical constraints that we started with. One of them was the question of social value, and I think the answer over here was, you know, if the people in Mali are going to be the ultimate beneficiaries of having a vaccine of this type, then there might be a reason that in response to that need, we do experiments there. This is interestingly countered in the debates with the concern about exploitation. So you're developing a vaccine in the United States, and you bring it to Mali to test it first in human. And there's been a lot of resistance to that historically, that products developed in the United States should be first tested in the United States in phase 1 trials, and then sometimes repeated in phase 1 trials in another country for the reasons of differential risks or differential genetics or whatever. Also questions about risks and safety. Are there risks to people in one location or another that might predictably be worse from the construct, the vaccine construct that was being tested? Not only risks that they might experience, but facilities to treat the risks that they may experience. One of the things that apparently was a potential risk from this vaccine candidate, which I think is true of most vaccine candidates, is the potential for anaphylaxis. And the question was, are there facilities set up to be able to handle anaphylaxis if this was an issue in Mali, for example? Huge questions about acceptability. Even if you're right, that it doesn't matter from an ethical perspective or a scientific perspective, it would be equally okay to do it in both locations. There's still questions about whether or not the review groups that reviewed the study would find it acceptable in their context. Now that is a sort of slightly different reason in a certain way, but it's an important consideration in terms of figuring this out. And then, of course, informed consent is always an issue everywhere, and the question is, in this kind of a context, is there an expectation about a malaria vaccine in a setting where malaria is an endemic that distorts the possibility of understanding the decisions about participating in a phase 1 study? Or is there a problem in the United States because people who volunteer for phase 1 vaccine studies, healthy people, are paid? And, you know, is there something about the money that distorts their ability to understand the risks? So there are a lot of interesting complications in terms of thinking through this in more detail. This slide is, again, lots of stuff on it, but I just want to raise it for a different reason. So I've given you some ideas about some of the complexities in thinking through study design issues, fair subject selection issues. Now here's one. This is just a proposal for a relatively straightforward study of diabetes, adults and children, some with diabetes, type 2 diabetes, and some healthy match controls, matched by a number of different things. And here's the sort of list of procedures that are involved in this study. And there's no benefit, because this is not a treatment study. People are going to be offered money, but there's no other benefit. So I put this up here only to ask, if you are evaluating these risks, would you call this a minimal risk study or a greater than minimal risk study? Yeah?
I think greater than minimal risk, because you've got children involved, and there's all kinds of things going on. Also,
Okay. So greater than minimal risk because there are children is what you said?
Partly because they're children.
So these things are riskier in children than in adults? Is that what your reasoning is?
The issues with getting consent from children.
Okay.
I know parents are supposed to give consent, but I think.
Okay. So there are issues getting with consent from children. And so this, I really appreciate your answer, and it's really the reason I wanted to put it up here, because there's so many layers of thinking this through that are important. Absolutely, consent from children in research is very different than consent from adults, because children can't give consent. There's compensation involved. But one judgment that needs to be made that's independent of the compensation and independent of the consent is how much risk is here. And each of these by itself, not everyone would agree, but I would argue that if you took each one of these by itself, it's probably minimal risk. But the interesting question and problem is, for all the reasons that you said, it's not that simple. You have to put them all together. The study requires all of them, and it requires all of them in children, and it requires all of them in children who are sick, and it requires all of those in children who are healthy, so, and adults who are sick and healthy. And so all of that together helps you think through.
So it seems to me that the riskiest thing in here is the total body MRI. I'm trying to figure out why in a natural history study of this nature you
Right.
want to do it. It's like a massive fishing expedition,
Right.
and there's no objective in mind.
Well, there might be, and I didn't write it down, and I can't tell you what it was either. But there might be an objective. There might be a good rationale for a full body MRI. I just don't know what it is.
In particular, to have a total body MRI,
Right.
or for that matter, anybody.
Right.
Has, because,
Can't-
I mean,
doesn't
that's
MRI
a
show
major
fat?
event.
Doesn't the
Yes.
MRI show the fat level?
So the response over here was maybe it shows fat, and so body composition.
You don't need an MRI for that.
Well, well, one of the good things about what you said is, when you're in the position of evaluating whether or not this is an acceptable list of risks for the population that's being proposed, one of the things, of course, you can do is say, it would be more acceptable if you took the MRI away. So, unless there's a really strong rationale for it, get rid of it. Yeah, sorry, go ahead.
In a study, how did they determine whether the risk is additive for each component of the study or whether it wasn't
Right. And they meaning, what do you mean by they? Okay. So the question was, how do IRBs and others determine whether risks should be additive or you should look at each line? Great question. I think there is no agreement on how that should be done. There are people who argue for what's called component analysis, which is looking at each independent procedure and saying, is this risk okay, is this risk okay? Which I think is a very useful exercise, but I still think my own view, and I share it with many others, is that there's a context that you can't ignore. And so the fact that there's 20 minimal risk interventions seems different than if there's one. The fact that there's children seems different than if there's just adults. So the context matters in many cases. So I think IRBs do it differently is the answer to your question. Jack?
I agree with you about the focus on cumulative burden as compared to what even the sick subjects would experience in their normal care.
Yeah.
But focusing on one thing, would you discuss whether in and of itself the DNA capture and storage of material for children indefinitely in itself poses more than minimal risk?
Yeah. I think the question was, does DNA capture itself and storage indefinitely pose minimal risk by itself? Take all the other procedures off the table. I think it depends on a number of things, like how it's stored, how it might be used in the future, whether anybody would go back and try to connect it with the child, what kind of promises were made in that regard. So there's a lot of important details that are, I think, necessary to be able to make a determination whether it's minimal risk or not. I know a lot of people would say it's greater than minimal risk from the get-go, but I'm not sure that I agree with that. Oh, I wanted to go this way. So this is another case, came from another location, and I think it's an interesting one. It's one that everybody's dealing with right now. And it has to do with informed consent. And I could have chosen, I could have given the whole lecture on informed consent. There are lots of different questions we could ask about informed consent. But this one is particularly interesting, that there are these interesting experiments that can be done with stored samples. And so in this case, somebody wanted to do a look at sort of precancerous mutations that were found in breast biopsy tissue and then correlate them with later development of breast cancer. And in order to do that, wanted to go back, a couple decades into the tissue sample repository and look at those in association with medical records. And the question, of course, is, can he or she, the scientist, do that? does the consent that the person gave, if they in fact gave consent for research at the point of having their breast biopsy done, allow it? And this is a really important question for today and yesterday, but it's going to be even a bigger question as we go forward for all kinds of reasons. One is that we're collecting tissues and samples all over the place. We have them everywhere. We have DNA everywhere. Second is there's all kinds of uses that we can put, important research uses that will serve to promote health that we could put to those tissue samples. And so the question of what consent should be on that decision is an important one, and one that's the center of lively debates, as many of you I'm sure are involved in. So I want to use that to turn briefly to talk about some other sort of developments in thinking about translational research. So Elias Zerhouni, who was the director of NIH in 2005, recognized that there's a whole bunch of tools that we now have, and he listed some of them here, that offer unprecedented prospects for advancing knowledge of human disorders in a translational context. And so, you know, there's a sort of sense of urgency here in translational research that maybe didn't exist prior. And we know that subsequent to that, we now have a whole institute that's devoted to translational research. And I think there's some really interesting projects that they currently have going on that raise ethical issues that have not yet been dealt with. So let me just point out a couple of them. Well, there are some things that are really good. So the Clinical and Translational Science Awards, as you may know, there's a network across the country of groups of people who are working on clinical and translational science. And one of the very, I think, innovative and important things that the NIH did was require there to be an ethics person in each of those CTSAs. So there's a research ethics resource for every CTSA. And that, to me, is thinking ahead in terms of some of the issues that people are going to have to deal with. Some of the rare disease issues raise interesting ethical issues, that some of which are common to the common diseases and some of which are very unique in terms of incentives and recruitment and advocacy and a number of things like that. I think there's a lot of issues in here that we're just beginning to think about. So one that I am aware of is this the issue of repurposing drugs and sort of the ethical considerations that come up with the process of repurposing drugs. So these are drugs that are available, that are used out there for other things and now being tested for a new indication. And, you know, how do you deal with that in terms of thinking about availability, informed consent, lots of really interesting issues in terms of repurposing drugs. Also, we know that there's been a lot of effort and a lot of excitement in translational urgency, as this article says, from stem cell research. And so this was a paper last year by Mahendra Rao and Francis Collins talking about the fact that there's been so much progress made in stem cell biology and the possibility of regenerative medicine, that there's this new translational urgency, and that we're now thinking about using embryonic and induced pluripotent stem cells and technologies to produce therapies and other treatments in the near future. This is something that's been on the horizon for a long time, but now we're like poised to do this. And this raises some really interesting issues as well. So one of the, I think this cartoon is pretty telling, but an article that was written by an ethicist at the Cleveland Clinic about this says, discussion of the bioethics of stem cell research has transitioned from controversies over the source. So a lot of the debates over stem cells over the last 15 years or so have been the source, the embryonic stem cell coming from embryos, and that hasn't gone away. There's still controversy over that, and there are a lot of rules and guidelines that are important to know about when doing stem cell research. But a lot of the focus has now turned on the use of stem cells, how we're going to use them both in research and in clinical medicine. And I think this is an important transition time for research using stem cells. One of the interesting things that this article that Mahendra and Francis put together was a list of what they call roadblocks. And I just circled a couple of them, but I think those are the most obvious ones. But when I look at this list, I see ethics almost in every one of them. There are roadblocks all along the way, and a lot of them have to do with ethics. They have to do with regulation, standardization, incentivization, business models, etc., consent, of course, risk management, lots of issues, all of which have ethical considerations. So I'm going to just give you one case to think about, and that is the case of a pretty simple one in some respects when you think about that long list in the previous slide. But this is the case of what should people be told when their tissues are going to be used for induced pluripotent stem cell research. And there is a recognition that, there's been a lot of discussion about the ethics of research with pluripotent stem cells in general, cell lines, specimens, genetic research, reproductive research, and transplantation. The interesting thing about iPS cells is, as noted by others, they encompass all of these. All of this is rolled into one. And so you have this interesting challenge of saying, okay, today I'm going to ask you for a donation of your cells. What do you need to know to be able to, in an informed and voluntary way, give that donation, not being able to predict precisely how those cells are going to be used in the future, but understanding the sort of range of possibilities and maybe even having some options about certain uses that you would opt out of. And so this is a really interesting problem that's happening right now in our setting at the NIH. I forget the number they told me. There's something like 100 labs that are doing induced pluripotent stem cell research. So we're collecting lots of tissues from people right now to do this kind of research. There's some people in my group who recently published a template for informed consent, and they did it by not only sort of culling the literature on consent, but also interviewing different institutions that do induce pluripotent stem cell research to see what kinds of issues they thought were important, gathering collaborators across scientific fields that are working on this kind of research, and came up with a proposed template which they recognize is, a proposal, one that can be useful today, one that people ought to continue to deliberate about in terms of whether or not it captures the things that people should know, want to know, and later will wish they had known when their cells are used for certain things. So can you think of some things, if somebody was asking you today, what would you want to know about the use of your cells to make induced pluripotent stem cells? What would you care about?
I have a question before that.
Sure.
Are these samples related to the specific person that needs to always be recognized that this cells belong to this person? Can have a privacy.
Yes.
Number one, if that's the case. The other case is that if it's a matter of privacy and the cells are using discoveries and then manufacturing and then the commercialization comes in, for the profit of it, if the person needs to have any participation in it, is recognized. I mean, all of this
Okay.
adds up.
So two very important issues that were just raised. One is privacy issues. These are my cells, but will they always be identified as mine? The answer is most likely yes, to some degree. The second is commercialization. Will they be used for possibly creating products and things that will make somebody money? And the answer is probably or possibly. Will I get some of that financial gain? Unlikely. Unlikely, at least the way people are thinking about it today. And so one thing that in this template, for example, one of the proposals is to say these could be used for commercial purposes, and you won't be getting any of those profits. Anything else you can think of you would want to know?
I would not want it to be used for construction purposes, biotherapy.
Okay.
But it would be hard, you don't have to write it like you write a legal document.
Yeah.
That anticipate things in the future. You don't want to prevent something being used. You don't want to be too specific in how you use things you don't want to use.
Right.
You want to make sure that somebody advising you, whatever using the stuff, that make sure it's used
Appropriately.
appropriately.
Yep.
Who's ever they deciding that? Who's the leading ethics person? Forever.
So, yay. So, two things that were said, to repeat for everybody that's listening. One was there are certain things I wouldn't want it to be used for, like bioterrorism kinds of things, and it depends on sort of how the uses roll out, and there's certain things that I would want to know about or maybe be recontacted for. So one of the things that the template again suggests is that there be a method offered to people for recontacting them. How do you want to be recontacted and included in this deliberation or conversation over time? There's also some information about who would be watching, who would be making sure that it was that the uses of these cells would be beneficent. And the answer would be there needs to be some oversight, whether it's an IRB level oversight or some institutional oversight on the use of cells. That makes sense, and I think a lot of people would want to know that. One of the things that they proposed and had a lot of debate about was whether it should explicitly say we won't use your cells to clone somebody, to make a new human being. In raising that, it raises a lot of issues about, whoa, you mean you could do that? There's this sort of interesting, but that might be something that people care about, right? And that's something that maybe it should be in there.
If the human race was at risk, you might not want to prevent that because maybe you need to do that to save us.
And that's,
Or you need an ethics person.
okay. Well, and there you need recontact too, right?
Yeah. Well,
So,
if the tissue donor is still alive.
yeah, okay. Yes?
Don't patients that enter NIH research protocols enter into a contractual arrangement to these issues are addressed on a protocol by protocol basis? What we can do with the data, what we can't do.
So the question was, when people enter NIH research protocols, do they enter a contractual relationship that spells out what we can and can't do with their samples? Well, two things I want to respond in that way. Certainly, everybody who enters into a research protocol at the NIH is given information about what the studies about that they're in and what the plans are for using for procedures that they might be asked to undergo, for interventions that they might be asked to undergo, and for using any samples that they might contribute. So the answer is, at some level, they're given that information. Most people, and there's lawyers in the room that could help me here, most people don't think of that as a contract. It's an informed consent process and document, which, has some elements of contractualness to it, but it's not a contract, it's not a formal legal contract in that regard. It's, yes, but not with the legal standing of a contract. And then, but this issue, and the reason this is an interesting one, is the problem in terms of being able to tell somebody today, this is what we're going to do with your cells, this is how they're going to be used, and we will or won't do X, Y, and Z, is that we don't know. That the science is changing so fast, and you don't want to create induced pluripotent stem cells and then not be able to use them because, like the slide before, the person didn't give consent for a certain uses of the breast biopsy tissue, something like that. Yes?
If this doesn't have the force of contract, this consent form, and you have an immortalized cell line, which is then passed to many, many other people through a long chain, and they may or may not adopt the same consent guidance, or even if they do, if they don't abide by it, are there any recourse to up the line to all the people who passed it on back to the original person? or is this a freebie?
So the question was, you know, lines that are made or passed from researcher to researcher, and is there any recourse for a researcher who doesn't follow the sort of terms of the agreement? Is that a fair restatement of your?
Is the passing of the line subject to the signing?
Yeah, yeah. So good question. So that's another issue that people have been very careful about trying to put in consent forms these days, that your data will be shared, that these lines will be created, that they'll be kept forever, that they'll be shared with other researchers and used for lots of different things, and that somebody will be watching, hopefully, to make sure they're used for beneficent purposes. Whether or not there's a established recourse for somebody who down the line breaks that sort of promise, I don't think so. Whether there will be someday, you don't think so. I don't think so either.
About
Yes.
future use of cells.
Yes.
It's like a will. That's why we have lawyers to break the will.
Well, except consent documents are not, I mean, that was part of my response here, and they're not legal contracts. So they're not like a will in some respects. They are like a will in terms of me saying, this is what I want you to do with my stuff, with my cells. Yeah, they are like a will in that regard. Yes?
My immediate reaction would be to one as restricted a consent as possible with re-consent. For example, the possibility that someone will decide to sequence the genome because they find my cells are doing something interesting.
Mm-hmm.
But that doesn't affect just me, it affects my kids and my relatives. Yes, they should have to come back to me to get that information if they want it, or my heirs with 20 years ago.
Yeah.
I'm wondering in general, what do these consent forms, do they tend to be more open-ended, favoring the researchers to do as much as they want, or are you trying to restrict them, or is it on a case-by-case basis?
Well, it's really changed over time. There are people who argue both sides of the way you just proposed it, whether there should be a sort of broad consent for lots of possible future uses as long as there's somebody watching, versus you can only do this, and anything else you have to come back and ask me. And there are trade-offs. There are trade-offs to both of those, for sure. There is not consensus on the way it should go at the moment. Yeah?
I think the biggest question for me is that even if somebody's watching, ethics is a very questionable word because it is that time.
Right.
And what is ethical right now is not what is ethical tomorrow or 20 years ago.
Well, so the comment was, what's ethical today is not necessarily what's ethical 20 years ago or tomorrow.
The progress and because what we need to do.
Well, I think there are some universal threads to the ethics that will persist through time, hopefully, and through different situations. But the interpretation of what they mean in this context, for this study, for this scientific experiment, is difficult, and it's not. It cannot be, I would argue, always the same. Yeah.
Christine, once upon a time, physicians in academic training were encouraged to do self-experimentation.
Yes.
In fact, many of the so-called great advances, this is up until I was in 70, there, post-war, experimentation of some horrendous, but others really quite remarkable. What's the current ethical perspective on that?
Yeah.
Self-experimentation.
I don't think there's any, I'm pretty sure there isn't any clear guidance, like there's not a policy or a regulation or a law about this. However, here's the way it seems to be understood. There are a lot of people that believe self-experimentation is perfectly acceptable, ethically, if there are the usual oversight and safeguards put into place over the research that everybody would get. So in other words, if you buy the framework that I gave you before, there's a reasonable research question, a good design that's valid and will get an answer. There's fairness in terms of selection. That may be a little trickier in this case. The risks are minimized, all that stuff, independent review. The independent review would consider the possibility of the investigator, him or herself, or his or her family, or his or her laboratory participating in the study. And there are some really interesting issues around that. Certainly the person who's the PI might not only understand the science the best, but be in a position to say, well, this is my choice. Once you get family members, laboratory members, friends, that gets a little trickier because people have reasons to be, whether explicit or implicit, feel a little pressure from, oh, well, this is my boss or this is my friend and they think it's so great, I should participate too. So it gets a little harder, and so there are lots of interesting debates about what safeguards should be in place to allow, for example, employees to participate in research. And there are some policies about that, but no rules, no regulations. Let me finish because I have a couple more, one other interesting issue that I think people would like to think about, and this is the issue of incidental findings. And I love this cartoon because I don't know if you can see it, but Kermit there has a hand inside of him, and the doctor's saying, what I'm about to tell you might come as a shock. So this is sort of an interesting, incidental findings are not new. In medicine, in research for many years, we've been finding things when we're doing research, finding things that we're not looking for. And the ethical dilemma has always been and continues to be, what do you do about them? Do you follow up on them? Do you try to figure out what they are? Do you tell the person everything you find? Are there some restrictions on what you tell? It's a huge debate and lots of interesting questions. And it has gotten, in the very recent times, much more complicated. And part of the reason it's gotten much more complicated is because we're using now next generation sequencing, whole exome sequencing, whole genome sequencing, where not only are there going to be lots of incidental findings inevitably, but it's even hard to know whether we should call them incidental anymore. There's this, you're going to get an entire genome and you're going to find lots of things. And so the question is, what do you do about them? So there was an interesting case that was published, just in June of last year. This was from a group here at the NIH. They were doing a whole exome sequencing, and this is just one pair of twins, so two people, two, in this case, children, one of them affected with the disease. And they did the whole exome sequencing, and they identified 79,000 genetic variants. Okay, once they cleaned them up a little bit and excluded things that were known, they found 32 novel variants in 32 genes that might be associated with human disease. So how do they deal with that? They were able to determine that 18 were associated with recessive disease, 18 with dominantly manifesting conditions. But here's sort of the punchline. In this case, I think luckily, they thought about it before they did it. They thought about, okay, what are we going to do when we find lots of variants? What are we going to do with them? What are we going to decide to tell people about? And so they had previously discussed this with their IRB, and their IRB came up with a criteria for return of information to the research participants. And so in this case, going from 79,000, there was only one that they ultimately felt they needed to inform the children, their parents, about. And so I thought it would be interesting to look at the criteria that this particular IRB laid out. They do make some sense. Some might argue they're too stringent. Some might argue they're not stringent enough. So I thought I would ask you what you think. So here are the criteria for return of information using next generation sequencing. The genetic change must be known or predicted to be of urgent clinical significance. Knowledge of the finding must have clear, direct benefit that would be lost, excuse me, if the diagnosis was postponed, made later. Potential benefit of knowing that a genetic disorder exists outweighs the potential risks of anxiety and subsequent medical testing that could result. Very interesting idea. A little bit hard, I think, to figure out how you would know that in advance. Unless they add substantial risk factors for multifactorial disorders are not reported. So if they find something that's, got lots of other factors involved, they wouldn't report it because it's too speculative, I guess. And then they have this really detailed discussion of recessive mutations, when they'll report recessive mutations. So if you look at this, and I know I'm sort of throwing this at you, but is there anything that you think should be added to this list or taken off this list? Yeah.
I applaud them for considering and getting back, but at the same time, I don't think they go far enough.
Okay.
And I think there should at least be an option that if the patient wanted it, or the people who were sequenced wanted it, that they should be allowed to get all the information. And
Okay.
EIH owes them to put them in touch with genetic counselors, make sense
Okay.
of the information as best as possible.
So the comment was they didn't go far enough. They should have allowed more opportunity for the participants to decide whether or not they wanted everything and set up resources for them to be able to figure out what everything meant. Okay? That's a understandable direction that a lot of people probably would agree with. It's a huge task.
It's a huge investment, but if we're going to be using these people, I think we owe them.
Yeah. One might come back and say, if that's the standard, maybe we won't do whole genome sequencing because we won't know how to deal with that and we won't have the resources. That's just, I don't know that that's going to happen, but it could be, yeah. Yes?
How do you deal with this kind of information on consent?
Yeah. Good question. I don't know what the consent form looked like for this particular study. I have to say that right up front. I know that especially the studies that are being done intramurally with whole genome sequencing, the IRBs are very attentive to what is said in the consent form about what findings will be returned and what kinds of incidental findings will be reported. So they're paying a lot of attention to that, so I suspect that in this case they had some pretty detailed language in the consent form, but I didn't see the consent form, so I don't know. Yes?
I understand some, if not all, ICs give patients all of their electronic health records if they request that.
Yeah.
Is that true?
Uh,
I know it's true in a couple of them.
It might be true. So the question was, do some ICs give patients their electronic health record if requested? It might be true. And yet, it also might be true that the genetic sequencing data is not in the medical record.
Yeah.
Yes?
I'm happy to see bullet point for what their first criteria for
Mm-hmm.
that's a very widely population.
Mm-hmm.
So because it's for the substitute, depending on where you conduct or meeting them,
Right.
that could
Okay. So the question was about the specific criteria for recessive mutations and when, and depending on who the couple was and how the gene came together for the two people. And you can tell I don't know the details of this genetics. So I think it's an interesting point. And what I would say about that is that the IRB, I think, was smart to come up with some sort of standard criteria. And because this stuff is so rapidly moving, that criteria should be thought of as criteria. And if there's a particular case where this criteria doesn't make any sense, that should be something that's discussed, preferably before the IRB, before the study starts, but if not, once they find it, and can make a case.
The reason I was thinking about it is, let's say you sequence a child and they have this recessive mutation that causes severe morbidity of mortality.
Right. Right.
But there's like 4, so you don't know who the genetic level is.
Right.
So, you could say maybe they find that they're more curious or people find
Yeah. 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 So it's a very important point. So it raises a different issue, and I'm going to come back at you with a different issue. So the question, I mean, the comment was, if you have a child who has a recessive gene, they may be fine until they meet a mate and have offspring, and if their mate has certain genetic characteristics, they may have offspring that are very at risk of serious disease. So that raises the question, it raises a couple questions. It raises the question of, what do you do with data of this type that you have for children? Do you keep it for a long period of time? Can people come back to it later and ask questions of that nature? I mean, let's say everybody agrees, let's not tell the child now because it doesn't mean anything. But it would mean something if they were, you know, later finding a mate, as you said. Should they be able to come back and say, well, did you find anything that I should know about now in terms of my reproductive decisions? And it's a really interesting question. I think people have struggled with the answer because, you know, well, you can probably imagine some of the reasons. I don't think anybody's reached any consensus on how long we should keep stuff available to people to be able to come back to, or whether somebody should be looking at it every so often because there are new findings, and now we know something that might be relevant to this person that we didn't know at the time that we did the study. That's another question that people keep asking.
A practical consideration about these agreements is very complicated and very wordy, and it can be treated like a car rental agreement.
Right.
Nobody reads it.
Right. Right.
Right.
So this was a comment about the practical reality of making agreements that cover all the bases, but that people will actually want to read. And the example was a car agreement. And you know, I think it is a constant challenge in informed consent in general. How to package the information so that it's as informative as it can be without turning people off to reading the document. And I think probably everybody in the room can relate to this. There was a study recently that looked at how often people read the agreements that you get on the computer when you're doing something, and you have to agree before you can go to the next screen. And I forget the number, but you know, it was like 2% of people read them. Sure, people have reasons that they want the results.
Isn't that why they use small print?
Maybe. There was another, yes.
I just wanted to comment on the thing about when you tell children. It's almost like you create another entity, like a trust. A trust is set up that only gives principles to the kids when they reach a certain age.
Yes.
You almost have to set something up like that to read genetic information for the child.
So,
That would be another
yeah,
layer.
this is an idea about setting up some kind of a trust for a child when there's genetic information being kept over time so that there's a way to access it later on. You know, I think, again, this is an issue that's not going to go away because more and more people are going to get sequenced one way or the other, and we're going to have data out there. Some people are going to keep their own in their computer at home or in their pocket and have to figure out how to deal with it that way. In other cases, it will be in the, I can't remember if I had one more slide. I think I'll skip that one in terms of time. I think I'll skip those. I just want to end with this one. This is just from the NIH website, and actually, see, this shows my technological inabilities. I tried to put one little video excerpt in here from a participant in research that I thought was quite compelling in terms of her saying how important research was to finding ways to understand and treat. In her case, it was Parkinson's disease. But I wasn't able to do the embedding of the video, so I apologize for that. So second best is pointing you to this NIH website, which actually does have a series of videos from participants who talk about their own view on what it's like to be a participant in research and how important it is for them to be able to have research done. So with that, I have no more formal comments, but I can still have discussion or questions if there are any more. Yes?
I have a question about the sort of the fruits of the research.
Yes.
We have all the ethics and informed consent during the research. The research is now concluded
Yes.
at the first stage, and it's passed on to some other bring it further.
Right.
Though, is there anything in this first group of researchers who try to ensure that the second group who take the fruits of that research use the same ethical guidelines? Or
So the question was,
Werner von Braun sort of
yeah.
situation?
So the question was about, the spectrum. So you start with somebody doing the research at the first stage, it gets passed on, so to speak, handed off to some other group of researcher who's going to take it the next stage, and is there any responsibility on the part of the first group for how it's ethically handled in the second group? Is that what you're asking?
Is that part of your ethics of the first group? That you'll only pass it on to people who have as least good ethics in the next phase?
Yeah, well, I think it depends on what you mean by passing on. So,
publishing.
Yeah, so this is what I mean. I mean, if you do good research and it's transparent and accurate and honest and all those things that we talked about before, and then you publish it, anybody who can read the paper can use those results to do what's next. Now, that's why it's important to remember that there are common goals and common ethical norms and common scientific norms across the spectrum, so that if everybody is sort of expected to follow those norms, then the next group who does whatever the next right research is based on those results would follow those same norms. Does it mean that there aren't bad apples out there? Of course not. I mean, there are. I don't see how the first group is responsible for later bad apples. How could they be? However, it does raise the interesting question, the H5N1 example. The question really was, is it a big enough concern about the possibility of accidents or bad apples that you shouldn't even do the research? That was the question that people were struggling with. Are the benefits of doing the research in terms of what we would learn for surveillance, for potential vaccine development, things like that, do they outweigh that risk of once you publish the results that this mutation can be done and the methods are in the paper, somebody taking it up and doing something bad with it? That's really the debate. And I think the way it's come out was, the NIH anyway has decided they're going to make some of those determinations up front. And if they, and there's a couple of levels of review in that process that they've spelled out, if they determine that there are other ways to get the valuable information, or the risks of doing it can't be mitigated sufficiently for whatever reason, then I think the decision will be the NIH will not fund that research. Doesn't mean it won't be done by somebody else, but it won't be funded by the NIH.
And clearly there's a lot of classified biomedical research that goes on.
Yeah.
And where it is passed on to other groups, but only select other groups.
Right? Yes?
Before you ask the question about the ethics of using stem cells, you asked another ethical question that we didn't explore, namely the reuse of data that was collected in relation to research.
Right.
Do you have an opinion or?
Do I have an opinion? So the question was, the slide I put up about using samples that had been stored from a decade or more ago, and medical records that were a decade old, let's say, and what kind of consent is necessary to be able to say we can ethically do that research today. That's the question I posed. I think, the particular case that I put up there didn't, at least for me, didn't give me enough details of what the consent situation had been. There are at least 3 scenarios that I can think of. One, there was no consent at all. These were specimens taken in an OR, stored in a clinical environment, de-identified, and sent to a repository. Now, in that case, the particular study wouldn't be possible because you wouldn't be able to connect the medical record with the particular specimen, right? If they were de-identified. It's possible clinical specimens were collected, stored somewhere, and not de-identified and hadn't been used for research yet, let's say. In that case, I think the person who said before, maybe recontact. If there was no consent at all, maybe recontact is the right way to go, and if you can't find the people, then maybe...
You just said a key word, de-identified. If the data is de-identified, what would be wrong ethically?
so this is a big conversation, but the way the ethics of using stored samples has been thought of over the last probably decade or two is that a lot of it rests on de-identification. So if you can take a sample and use it for research and there's no way to connect it to the person from whom it came, then there's no risks to that person. And therefore, research can proceed as long as it's good research and, you know, all those other things.
Maybe something softer, not the samples, but the results, the analyte values, the clinical data is well collected.
Right.
And you're not touching any of the specimens or anything, but you've got all this data. It seems reasonable that you de-identify.
Right.
That no one who uses it to do research will know which are
Right. Well, that's the way it's been, yes. There's no problem from an ethical perspective. There are people who quibble with that, I'll just be honest.
Jim Sabino just announced that we can do that now with
We can.
data.
Yep.
De-identify.
Yep.
So others can get to it.
Yep. So Beatrice, for those of you who are listening, the comment was that the clinical center has a system called Beatrice, which is a repository of clinical data collected over many decades and is now available for research projects using de-identified clinical data. And some very important projects can be done with that. And the analysis has been that data that's de-identified, can't be traced back to the source, can be used in an ethical manner for good purposes without any risk to the source. And that's the way it's been handled.
I have another question, but I don't want to.
Yeah. Go ahead, unless somebody else has a question.
Okay. I have understood, maybe improperly and incorrectly, the distinction between translational research and translational medicine as being the following. Translational research, in my mind, is how can we translate this research from this lab to the next lab to the next lab. In other words, keep advancing the research, which we do a lot of at the NIH. And translational medicine is more this bench to bedside thing. How can we take the research and actually, you know, from the results of that research, convert it into products, diagnostic tools, medications that are going to immediately provide health care to the patients. Does that make sense?
Okay. So the question was, is there a distinction between translational research and translational medicine, such that translational research is just one lab to the next, translational medicine or something like that, and translational medicine is the more bench-to-bedside model?
Yes.
I think of the bench-to-bedside model as translational research. I'm not sure exactly what you meant by the first category. I think translational research and translational medicine are sometimes used interchangeably. I think the way I understand it, and I think one of the slides I showed earlier is that a lot of people have different meanings when they say translational medicine or translational research. The one that makes the most sense to me is the idea that some research, biomedical research, starts with an idea, an experiment in the laboratory, and goes through a number of important steps along the way, with the ultimate goal always being finding something that's useful to improve health in people.
Certainly that's very clear, but I guess my question is, do we have any mechanisms or processes, IRBs or scientific directors, that sort of push that, that really make sure that the research is actually going to be getting out in the real world and making a difference? Because I see so much research
Right.
just doing the same stuff because that's what they enjoy, and I don't quite see that we have a motivation to get it out the door as products that are going to improve health.
Well, so the question is, are there people who are pushing
Yeah.
for getting it out the door in a way that will improve health?
Yeah.
I think the answer is there are people that are pushing. Whether or not the processes are in place to make it happen for a lot of the research that's done at the NIH is a different ballgame, I think. I think there is a commitment to this idea of translational medicine or research, bringing it to the point of products or improvements in health in one way or the other. But as the IOM pointed back in 2003 or 4, there are significant blocks to those translations. And they're not only, I guess, things that happen at the NIH. There are ways, I mean, there's been some really interesting work, for example, recent work, and maybe longer than that, but I'm more aware of it recently, work where there are clinical research findings that are significant and might, if adopted, change practice, and people don't adopt them. Practitioners don't adopt them. And so the question is, why? And so there's a need for addressing that translational block as well. There's absolutely a need. There's clearly need for understanding important, significant findings that we can determine in clinical research settings, how we can put them into implementation packages that work in different settings. That's a similar but different question than the previous one. So you're right, there's a huge amount of need in that effort. There are people who are doing that. Some would argue, many would argue, there's not enough attention to that kind of research. There's not a good funding source for that kind of research. You can do the experiment, because I've done it myself, where you go around NIH and you ask people, do we do implementation research? And you'll get some people say, absolutely not. And other people say, oh, yes, of course we do. And then I suppose the next question would be, well, what do you mean by implementation research? You know, so there's a lot of, yeah.
Okay.
Other? Okay, thank you. Thank you very much.
Very, very much.
You're welcome.
It's very good. You know, I would point out that this course is a small part of this communication, which is the key on which all this translational, no matter which direction it's going in, from the more bedside to the bench or the other way around, you have to be able to have some appreciation of the language, the art, the communication. It doesn't happen automatically, even if you throw a lot of money at it. Thank you very, very much, Christine. That was very exciting.